Hi, everyone. It's a pleasure to introduce Craig. So Craig is Canadian, as I just confirmed through Tanya, who asked directly. Craig did his PhD at University of Alberta, and he did both biochemistry and NMR to study synthesis of ubiquitin chains and the role in DNA repair. And after that, Craig came to Stanford to do a postdoc with Fordyce and also Daniel Herschlag. So there he wanted to keep studying the kind of the structural mechanisms of enzyme function, but as we all know, to really do that, we are still lacking the perfect tools for measuring enzyme kinetics in high throughput. So Craig decided that he's going to be that hero who's going to make that method work. And that's what he was working on during his entire postdoc. So I met Craig for the first time about a year and a bit more ago, before the pandemic started, when we could still visit each other. So I visited Polly's lab, and I was really impressed when Craig showed me his results. And then in the meantime, his preprint came out, and I was impressed again. So now I'm ready to be kind of impressed for the third time when I see Craig present everything in a nice way. Just for the technical part, please, if you have any questions, we encourage discussion, and put your questions in the Q&A box, and I'm going to read them in the end, and Craig's going to answer them. If you insist on asking your question in person, please raise your hand, and we're going to unmute you so you can ask Craig your question by yourself. Okay, so let's start. Thank you.
Everyone can see my slides. It's good. Great. Thank you so much, Tina, for that introduction, and hopefully I can live up to it. So today I want to tell you about work I've done in collaboration with a really amazing team of biochemists and bioengineers in developing a new technology, and really I think a new approach to studying enzyme function that I believe will ultimately and hopefully transform our ability to study, understand, and actually eventually design enzymes. So enzymes catalyze and control all of the reactions of cellular metabolism. So understanding them, of course, is critical for understanding biology, and especially disease and disease progression. Of course, equally importantly, enzymes are among the most proficient and specific catalysts known. So if we had the ability to engineer new enzymes and do this efficiently, it would be transformative for not only medicine, but for industry as well. So actually, the sort of potential power of being able to engineer novel catalysts was recognized very early on in the study of enzymes, and actually even before the first crystal structures of enzymes were solved. For example, in 1955, Linus Pauling suggested that when our understanding of enzyme activity becomes great enough, it'll be possible to synthesize a catalyst. So how are we doing? Well, in the intervening 70 years of study, we've solved the structures of thousands of thousands of enzymes and enzyme mutants. We've made and assayed millions of site-directed mutants and elucidated the chemical mechanisms of many, if not most, of the reactions carried out by known enzymes. And true to Linus Pauling's prediction, we have engineered a few de novo enzymes. But there's a problem. And the problem is that the computationally designed enzymes are far, far less proficient than their natural counterparts. And that's even after multiple rounds of directed evolution. So we're clearly lacking some sort of fundamental and practical understanding that's preventing us from being able to efficiently engineer enzyme catalysts with proficiencies that match those that have evolved naturally. And this is because all of these studies have tended to focus on the active site alone. Now, of course, this makes sense. The active site is where the substrates bind and where they undergo their catalytic transformations. But we know that active sites don't function outside the context of the folded enzyme. And this context, illustrated here, consists of this dense thicket of hydrogen bond, electrostatic, and hydrophobic interactions. And what's missing from our understanding has been an understanding of this context on the function. And to understand this context, what we really need to do is to make mutations. We need to vary it. And we need to make lots of mutations because enzymes are large. And then we need to observe and interpret the effects. Now, more modern high-throughput approaches can look at mutations beyond the active site. And these experiments do often tell us that mutations throughout proteins and enzymes frequently do have functional effects and large functional effects. So for example, the function-enhancing mutations in directed evolution experiments are often found far from the active site. But directed evolution experiments typically only pick winners so that we don't get or we lose the functional information on the defective mutants. And that's the information we could use and synthesize to be able to more efficiently design catalysts. And even when we do want to functionally characterize the winners, that often requires more low throughput studies as well. Now, deep mutational scanning experiments have the advantage that they can look at mutations everywhere and we get all of the information. We're not restricted to the winners, but these experiments typically measure fitness and not activity directly. So they're measuring the survival of a cell that's expressing the protein or enzyme, for example. So we're not looking at catalysis directly. And the issue there, and just to take an illustrative example, let's imagine we make an enzyme mutant that has a 10-fold drop in the rate of product formation. And that's sort of illustrated here in red. Now, this 10-fold drop might be due to a catalytic defect in the mutant enzyme, but it could just as easily arise from a destabilization effect on the folded enzyme. So rather than a catalytic effect, our mutation could simply be resulting in a significant fraction of unfolded enzyme under the conditions that we measure. And these two mechanisms would be fundamentally indistinguishable from a single progress curve. To further illustrate the point, it's also possible that the mutation doesn't have to affect the catalytic efficiency, the Kcat over Km directly. It could simply be changing the microscopic Michaelis-Menten parameters, in this case, Kcat and Km. Now, if we assay these two enzymes at high substrate concentration, they're going to look really, really different. But if we assay them at low substrate concentration, they're going to look nearly identical. So what we really need is a way not only to make a lot of mutants of enzymes, but we need to functionally characterize them across a variety of different parameters. We want to measure catalytic effects, folding effects if we have them, and other parameters which are sort of unique to different systems. So in other words, we need to do the assays of traditional biochemistry with all their quantitative rigor, but in higher throughput, or a scale that matches the scale of enzymes. And so, as I've said, traditional biochemistry is really, really powerful for making mutants and then assaying them across the complete functional vector across all these aspects of function, but it's pretty laborious. So for example, to express, purify, and then assay mutations at every position of a typical enzyme of a few hundred residues, this is going to take years, and it's going to cost a lot in reagents and labor costs. And probably most importantly, the person doing these experiments for those years is probably going to be pretty miserable during that period of time. So instead, what we've developed is a method for expressing, purifying, and then assaying thousands of mutants in a space of a few days rather than a few years, and with much less labor and vastly reduced cost. And this technology we call HT-MEK, which stands for High-Throughput Microfluidic Enzyme Kinetics. And HT-MEK basically does what it says on the tin. It's a microfluidic chip with 1,568 different microchambers. And in each microchamber, we're going to express, purify, and then assay, quantitatively assay a different enzyme mutant. So I'm going to tell you how HT-MEK works now. So in the first step, we simply assemble a library of mutants, a library of mutated plasmid DNA, into a series of 96 and 384 well plates. We then print a DNA array from these plates onto a glass slide. And the important thing here is that we know the identity of each mutation in the array so that throughout our experiment, genotype and phenotype are coupled. We then align our microfluidic chip on the glass slide over the DNA print so that each DNA spot, illustrated here as a blue circle, becomes isolated in its own microchamber. And our chip also contains a series of micromechanical valves, illustrated here in light gray. And these valves are going to be important for allowing us to control fluid flow through the chip and also for starting and stopping reactions. So in the next step, we want to express all of our mutant enzymes simultaneously. So to do this, we simply flow through a cell-free expression system, resolubilize our DNA spots, and let the protein express. typically by we just put the slide on a hot plate. And all our enzymes are C-terminally GFP tagged, so we can follow expression simply by imaging the whole chip in the eGFP channel periodically over time. And so we make a bunch of mutant enzymes, and then we want to purify them. So what I haven't told you about so far is that on these valves here, and hopefully you can see my mouse, we call these the button valves. And we're able to pre-pattern these valves with an anti-GFP antibody, so that when the enzymes are produced, we simply open the valves and let the enzyme diffuse over, and then they get pulled down or captured by this anti-GFP antibody. And sort of a blow up of this process is here. So these green spots sort of schematically illustrate eGFP tagged enzyme being pulled down to the anti-GFP antibody. And this is a valve that we can open and close. So once we've pulled down enough enzyme, or as much enzyme as we want, we simply close these valves. Oh, and I'm sorry. So this is just a schematic illustration of the pull down. So the surface chemistry is set up so that we pull down a BSA molecule, biotinylated BSA, recognized by neutravidin, which itself recognized a biotinylated anti-GFP antibody, which pulls down our enzyme. So we've made and pulled down our enzyme. Now we simply close these button valves, and that's denoted by the dark gray state. So when we close them, we sort of press the enzyme down against the surface of the glass slide, and this effectively removes the enzyme from solution and keeps it in place and prevents mixing between chambers. So we protect our enzyme, and then we simply wash through the device to remove all the cell-free expression mixture we no longer need. Now, of course, our goal here is to get kinetics for all of these mutants, and getting kinetics requires knowing the amount of enzyme within each chamber. So to do that, after the purification step, we simply image in the eGFP channel. And I'm showing here about 1/4 of one full chip, and you can see that we have eGFP tagged enzyme spots for wild type enzyme and a bunch of mutants I'll talk about later. Now, the important thing to know here is we have spots where we, in chambers where we've deposited plasmid, but we don't see eGFP spots in the chambers we've deliberately left blank. And this is telling us our valves are working, and we're able to prevent cross-contamination throughout this process. So the button valves, the valves in which our enzymes are surface immobilized, allow us to start and stop reactions as well. So to run kinetic assays, we simply close the valves, protect our enzyme and remove it from solution, and then introduce the substrate. So we just flow through, equilibrate the chip in one substrate concentration, and then we open the valves, expose the enzyme to that solution, and the reaction begins. And our substrate is fluorogenic, meaning that when the enzyme reacts with it, we generate a fluorophore. So to get kinetics, we simply track the buildup of fluorescence over time. And again, I'm showing you about 1/4 of the chip, and we can see lots of chambers lighting up quickly, which are wild type or wild type like mutants. And we see mutants of a variety of different rates coming up. So now I'm going to tell you, we have all these fluorescence buildup curves. How do we go from these fluorescence measurements to Michaelis-Menten parameters? So I'm going to show you one chamber and to illustrate the process, but remember we're doing all of this simultaneously. So in the first step, as I've said, we simply image in the eGFP channel. We measure the amount of enzyme immobilized on the button via a standard curve relating eGFP fluorescence to enzyme concentration, which we determine using a different experiment I won't tell you about. But for example, this spot here for this mutant corresponds to 4.1 nanomolar of enzyme in that microchamber. Next, we measure a fluorescence standard curve that relates the fluorescence of our product to its concentration. And it's important here to note that this is exactly how one would do a traditional biochemical assay in a plate reader or spectrophotometer, where you get a standard curve of fluorescence or absorbance corresponding to the concentration of the molecule. Having our standard curve, we can then introduce substrate into our device and then open the valves and let it react. So this is just a buildup of fluorescence over time, converted to product via our standard curve. And just as in traditional biochemistry, we fit an initial rate to this curve. And we, of course, we're doing this for 1,568 chambers simultaneously. So we've developed algorithms to sort of decide how many points are well fit to an initial rate. And we also have estimates of the background rate shown here in red. And these are important in later quality control steps. And so once we finish reacting one substrate, we simply reclose the valves, protect the enzyme, and then flow out the product. We can then iteratively introduce additional substrate concentrations on the same chip using the same enzyme and acquire initial rates of product formation at a variety of different substrate concentrations. We can then fit these and determine the gold standard Michaelis-Menten kinetics of classical or traditional biochemistry, Kcat, Km, and Kcat/Km. Now, we make all of our chips in-house, and it's actually pretty easy to do that. And it's also pretty easy to run multiple chips at a time. So we've made a lot of measurements. So to date, we've done about 200 chips, and that's comprised about over 670,000 individual time courses. And we've used these kinetics to determine 5,000 or over 5,000 kinetic and thermodynamic constants for a variety of enzyme mutants. And for the remainder of the talk, I'm going to be discussing the functional interpretation of these measurements and what they've taught us about enzyme architecture. But before I do that, I want to show you that HT-MEK works, that it reproduces the Michaelis-Menten constants of traditional biochemistry. So to do that, I'm going to introduce our model system that we use to develop HT-MEK. So we chose to develop HT-MEK using a bacterial phosphatase called PafA for a variety of reasons. One, it's mechanistically well characterized. So this enzyme has been studied extensively already in the Herschlag lab. Its mechanism is known, and the function of the residues at the active site are also known. So this provides an ideal jumping off point to move beyond looking at the active site and looking at mutations throughout the scaffold. Now, PafA is also one of the most proficient enzymes known with a 10 to the 27th fold rate enhancement. So this gives us a large dynamic range to explore when making mutants. And it's also a secreted phosphatase, so it's extremely stable. And we wanted to choose a stable enzyme so that we could focus on the catalytic effects of our mutations as much as possible. Now, I'll tell you more about the mechanism in a minute, but I'll show you now that HT-MEK reproduces the traditional biochemical constants. So to test HT-MEK, we simply measured the rates of wild-type PafA and 6 previously characterized active site mutants. And we can see that the rates determined from a number of replicates on chip, shown here in the box and whisker plots, agree well with the measurements made using traditional biochemical techniques, shown as gray circles. And importantly, HT-MEK has a greater than 10 to the 6-fold dynamic range in these parameters. So that's a big window for us to make mutations and quantitatively measure functional effects. So with this technology in hand, we're then poised to start probing or examining the functional context of mutations beyond the active site. And to do this as a first application, we mutated every residue in PafA to either valine or glycine at 2 substitutions with very different side chain properties. And then we just assayed all of these mutants on chip. So now I can tell you a little bit more about the PafA mechanism. So it's, as I've said, a bacterial alkaline phosphatase which hydrolyzes phosphate monoesters. Its active site contains a bimetallozinc core, as well as 4 residues, 4 side chains, which directly contact the substrate. Now, in the first step of the reaction, the phosphate monoester binds, and then a nucleophilic attack is performed by this threonine nucleophile, which is activated by the first zinc on the phosphorus. That results in the leaving of an alkoxide leaving group, which is stabilized by the second zinc. This generates a phosphoryl enzyme covalent intermediate. So in the second step of the reaction, hydroxide comes in and performs a reverse nucleophilic attack on the phosphorus, regenerating the enzyme and generating inorganic phosphate, which then diffuses off the enzyme. And it's important to note here that all of the contacts between the enzyme and the substrate are via these phosphoryl oxygens. There are no contacts between the enzyme and the leaving group. And this is a really cool feature because it allows us to use a variety of different substrates in our assays. And so, as I've said, we made every mutation to valine and glycine, and we began by assaying the effect of all of these mutations on Kcat/Km. Now, I've emphasized in earlier slides that it's reasonably easy to run chips and get lots of replicates, so that's what we did for each mutant. So for every valine or glycine mutant, we acquired between 3 and about 15 replicates. And this allows us to do rigorous statistical tests to assess the significance of effects or fold change effects from wild type. So that's what I'm showing with this volcano plot here. So I'm plotting the p-value for each mutant as a function of its fold change in Kcat/Km, catalytic efficiency, relative to wild type. And you can see that we can resolve about greater than 2-fold differences from wild type, and we're measuring mutations that are as much as 10,000-fold down. And really interestingly, when we do this analysis, we see that almost 1/4 or actually over 1/4 of all mutants reduce the K-cation of the enzyme. And as expected, the largest effects on catalysis are at or around the active site, which are shown here in dark blue spheres. But these catalytic effects extend from the active site throughout the entire enzyme. And this distribution is asymmetric, so we see regions of the enzyme sort of up front here with lots of catalytic effects. However, there are also different regions where we see almost no catalytic effects. Now, I told you the reason we did 2 mutations for every residue is because effects are obviously going to depend on the nature of substitution, and this is what we observed. So for example, for the polar and charged residues, shown here in red and orange, they can tolerate glycine substitutions reasonably well without a big catalytic effect. However, when we introduce valine, we see large catalytic effects. Conversely, if we mutate the hydrophobes in dark blue to valine, we see almost no effect relative to wild type, but these really don't like being substituted to glycine. But even though we see different actual residues affected by different substitution types, the distribution of effects for both the valine and the glycine scan are very, very similar. So now we're going to go further and measure not only the effects on catalysis, but measure effects on other functional parameters. So we know, of course, that active sites don't function in the without having the context of the folded enzyme, as I emphasized in my introduction. And we know, of course, that residues throughout the enzyme are going to be important for folding. So one of the simplest models is that maybe we're just making mutations and unfolding the enzyme. So to test this, we can actually just do a urea denaturation assay on chip for all of our mutants. Again, exactly as one might do in traditional biochemistry. So for example, let's assume we make mutations all over the enzyme with very different stabilities, or a range of stabilities. And these are just simulated curves to illustrate the point. So, okay, so now for destabilization to result in an apparent catalytic effect under our conditions of measurement, so 0 molar urea, this means that these mutations would have to be significantly unfolded at 0 molar urea already. So that's denoted here in the green curves. You see a variety of mutants that are 50%, 25%, and 75% folded. So these other mutations, while having a wide range of stabilities, are all almost 100% folded under our standard conditions. And therefore, even though they might be destabilized, they won't affect catalysis. So we don't have to worry about these. We only care about the green curves. And we can now easily test if we have any mutations in this regime, because these mutants are going to be expected to be extremely sensitive to low concentrations of urea. So we've measured everything so far under standard conditions, 0 molar urea. And if we add 2 molar urea, we'd expect that if we had any mutants in this regime, their rates would drop almost to 0, whereas the rates of these other blue mutants would be largely unaffected. So that's shown schematically on the left. So we expect, if we have very destabilized mutants, we should expect the rates at 2 molar urea to drop substantially relative to those at 0. So we measured the we measured the activities of our entire library of mutants in 2 molar urea and compared these rates to the rates at 0 molar urea. And we see that for all mutants, their rates are within error of this 1-to-1 line. And this is telling us that none of our rate effects arise from an equilibrium unfolding process. So all of our mutants are still highly stable at 0 molar urea. So at this point, we've discovered that over 1/4 to 1/3 of all of our mutants have a catalytic effect, and none of these are due to unfolding. But we actually suspected we might be missing some of the catalytic effects by using this fluorogenic substrate CMUP I've told you about. So CMUP, shown here in the top, is a great substrate. It's really bright and it provides a direct readout of catalysis because we just watch the formation of fluorescence. However, it's such a good substrate that the reaction with the wild type enzyme is actually diffusion limited. So that's illustrated by the height of this barrier. It's actually harder for the substrate to get onto the enzyme than it is to react once it's on. So in practice, what this means is that if we start making mutations with very small, subtle effects on the chemical step, shown here as the colored bars, if they're not deleterious enough to change the rate limiting step, we won't see them in CMUP hydrolysis. We'll still be measuring CMUP at the diffusion limit, and that's illustrated by this red line. It's only when we start making mutations that have more and more deleterious effects on catalysis that we actually resolve them using CMUP. So now in traditional biochemistry, we can get around this problem by using an intrinsically less reactive substrate. For example, an alkyl phosphate ester, such as methyl phosphate. Now, methyl phosphate is hydrolyzed to methanol and inorganic phosphate, and this reaction is less favorable, it's slower, and its chemistry is rate limiting for the wild type enzyme and all of the mutants. But of course, if we want to measure this on chip, it's going to be really hard in principle because neither methanol nor inorganic phosphate is fluorescent. And it turns out there's a commercial solution to this problem that we were able to adapt to work with our on-chip assays. And so you can buy this protein called the phosphate sensor from Thermo Fisher. And what it is just E. coli phosphate binding protein with a covalently attached coumarin fluorophore at the active site. So when this protein binds phosphate, the fluorescence properties change of the coumarin, and then we can simply measure this change over time to infer the rates in this very analogous manner to the direct readout of catalysis. And then importantly, this also makes HT-MEK more generally applicable. So now we don't need to worry about having bespoke fluorogenic substrates for all the enzymes we want to study. We can now measure the rates of any enzyme that generates inorganic phosphate either directly or via a coupled assay on chip. Okay, so we measured the rates of methyl phosphate hydrolysis for all of our mutants, and we expected all the rates of the mutants to follow this blue relationship, which we call the relative reactivity line. And indeed, if we make the active site mutants and compare to wild type, these rates do fall on this line. So for example, mutants with modest effects, such as T79S or N100A, they're still diffusion limited. So you can see we would not see any change in CMUP hydrolysis on the y-axis. It's only in the x-axis that we see a catalytic effect. When the mutations become more and more deleterious, we start seeing the effects on both substrates. So what we expected was that all of our mutants measured so far, or that I've talked about so far, would fall in this region here, to the left of the red dashed line. And we would, by measuring methyl phosphate hydrolysis, we would identify a whole bunch of other mutants that have more modest catalytic effects, but catalytic effects nonetheless. And this is in part what we did observe when we measured methyl phosphate hydrolysis for our full library. We see a whole bunch of mutants moving left on this relative reactivity line, sort of left of wild type, and that fits our model. However, we actually see more mutants either on this one-to-one gray line or in the intervening space. And instead of catalytic effects, what this actually indicates is that the mutants have similar effects on the rate of both substrates and that there are changes in the active fraction of enzyme. And I'm going to talk a bit more about this in a minute. But just to illustrate the point schematically, we expected all of our mutations to fall on this blue line. So any mutations that fall on this blue line have solely catalytic effects. This is what we expect. But we also observed a bunch of mutants that track on this one-to-one line. And what this means, according to our model, is that these mutants are wild type like, except there's just less mutant. There's some fraction of the total enzyme concentration that's just inactive. And then we also have mutants in the intervening space. And these are mutants that have both fraction active effects, but also catalytic effects within the active fraction. Okay, so this is our model. We have a model that some of our mutations have catalytic effects, but others are somehow partitioning into an inactive state. So what could this be due to? So you remember I've shown that there's no equilibrium unfolding. That was the purpose of a urea assay. But actually, another class of model is that there's a non-equilibrium process that's reducing the amount of active enzyme. So remember, we're expressing all these enzymes on the chip. So imagine if during normal folding, we make the nascent chain, it's coming off the ribosome, and it should fold correctly into the active enzyme, bind substrate, and then reactive product. However, what if instead of correct folding, these mutants are actually partitioning into a misfolded, kinetically trapped, catalytically inactive state? So at this point, it was just a model, but we wanted to test it. And it's actually sort of convenient that HT-MEK allows us to express all of the enzymes on the chip, because this actually gives us a lot of control over our expression conditions. So we realized we could test this model simply by varying the expression conditions. So there's a couple things we could change. I've told you that the enzyme active site binds these two zinc ions, which are essential for catalysis. So we simply reason that if we have more zinc present in the expression reaction, we should favor partitioning into the active folded state. If we have less zinc, we should favor partitioning into the kinetically trapped state. And we know from in vivo protein expression experiments that temperature can really affect enzyme and protein expression. So we also changed the temperature of expression on the chip, presuming that this might change the partitioning as well. So the experiments we did were pretty simple. For any condition, either zinc or temperature, and here I'm showing temperature, we basically just took two different chips with the same mutants and expressed one of them at 37 degrees, that's our standard expression temperature, and we expressed the other at 23 degrees, at lower temperature. And then after expression at different temperatures, we simply did the rest of the process, the binding, the purification, and the assay under the same common conditions. So just 23 degrees and 100 micromolar zinc. So we're changing expression, but assaying under common conditions. And this gives us a prediction. So if our misfolding model is correct, what we expect is that when we express at lower temperature, we should see the rates of a lot of mutants become faster than when they were expressed at 37 degrees, even though we're assaying under constant temperature. And that would be denoted by this right shift when we compare the two sets of rates. And that's exactly what we see. So we see that dozens of mutants are actually much more active when expressed at 23 degrees relative to being expressed at 37 degrees. And these aren't small effects. So in some cases, these differential effects are 10-fold, 100-fold, or even more than 100-fold more active when expressed at 23 degrees. And remember, this is telling us that our misfolded inactive state is long-lived and not exchanging with the active state. Because if there was exchange on the time scale of the experiment, which is typically a few hours to days, all of our mutants would be on the one-to-one line because they would reach a new equilibrium at low assay temperature. And we see similar effects in our zinc experiment. So when we express at a relatively high concentration of zinc, 1 millimolar, we see that the rates of lots of mutants become much more active than when we express them at a relatively low concentration of zinc, 10 micromolar. And remember, we're assaying again at constant zinc. So interestingly, if we take all of our positions that have temperature and zinc effects when mutated and put them on the PafA structure, we actually see that these effects cluster in different regions of the enzyme, but cluster together. So there's a region sort of behind this beta sheet that's very dense with temperature sensitive effects, and there are other regions that are sensitive to zinc effects. So this is suggesting us that there's specific interactions and specific contacts between residues in the PafA structure that are essential, not for catalysis, but just for getting correctly folded, getting into the active state. And so at this point, we said to ourselves, well, this is really cool, but we're expressing in these microchambers in this PDMS device. PDMS is super hydrophobic. So what if this is some sort of artifact of the chip? So we actually took a subset of these mutants and expressed them in cells at different temperatures and saw similar temperature dependencies expressing in vivo. We also ran native page gels of wild type and 4 mutants very prone to misfolding. And at 23 degrees, we see a distinct band for wild type in all of these mutants. But when we express at 37 degrees, we see that we still get largely one band for wild type, but now we get 2 bands for these temperature sensitive mutants. And we presume that one corresponds to the native active state and the other corresponds to the misfolded state. And actually, I'm not going to show these data in the talk, but the misfolded state is more sensitive to proteolysis than the active state. And we also have some experiments to show that only this band is active. Okay, so now we have a little bit of a problem. So, because I told you, remember in the early slides, I said we chose PafA because it's the stable secreted enzyme, and we wanted a really stable enzyme so we could only focus on catalysis. And although we don't have any equilibrium unfolding effects, what we do have are all these misfolding effects. And so these are convoluting our measurements of catalysis. So it's essentially a problem that boils down to having 2 unknowns. One unknown is the fraction active, the active fraction of the enzyme in the total concentration. And the other unknown is the catalytic effect within the active fraction. Well, it turns out that we have 2 unknowns, but we also have 2 measurements. We have CMUP hydrolysis and methyl phosphate hydrolysis and the relationship between them. So 2 measurements and 2 unknowns actually allows us to solve for the catalytic component of the mutation and the fraction active component. So for each mutant, having these measurements allows us to deconvolve the fraction active effects from the catalytic effects we're sort of more interested in. And when we do this for all of our mutants, and we see that the catalytic effects, again, And that's what I'm showing here. Only the catalytic component of mutations now. The biggest catalytic effects cluster in and around the active site as expected. But again, catalytic effects are still not restrained to the active site. They extend outwards asymmetrically throughout the enzyme. So now we actually want to even go further. We want to say, okay, we know we have all these catalytic effects, but can we actually ask, rather than just saying, oh, this residue over here has a 10-fold catalytic effect, can we actually try to determine the mechanistic basis for each catalytic effect? Can we start to test models for how mutations up and around here, for example, or distal to the active site are affecting individual elements of active site function or individual catalytic mechanisms used by the enzyme? And to do this, we've developed an approach which we call functional component analysis. And it's a method that provides a description of the functional architecture of the enzyme that's rooted in prior mechanistic insight. Essentially and simply, FCA involves measuring the effect of each mutation, not only on K-cat over Km for one substrate, but K-cat over Km for multiple substrates, K-cat for multiple substrates. And we also measure the binding of multiple inhibitors. And then we choose these measurements and make quantitative comparisons between them to determine the source of catalytic effects. So I'm going to take you through a couple of examples of what we do. So the first example we call functional component 1, which we refer to as effects through the O2 oxygen atom. So PafA has these two residues, K162 and R164, which sit at the N terminus of this yellow alpha helix. And these residues in the crystal structure appear to be donating hydrogen bonds to this oxygen. So this structural evidence actually makes a prediction that we can test. So if the functional role of these two residues, K162 and R164, are to donate hydrogen bonds to this oxygen, which we refer to as the O2 oxygen, we expect that if we assay a diester substrate, which has a methyl group on this oxygen, these two residues should provide no catalytic benefit for this reaction, because with the methyl group here, they can't donate hydrogen bonds to the oxygen. So Fanny Sundin, a research associate in the lab, tested this model using traditional assays. So Fanny mutated K162 to alanine, R164 to alanine, and both of these both of these residues together. And what she saw was that for each mutant, the rates of monoester hydrolysis, the rates of this reaction, dropped when she made mutations, which we expected. But the rates of the diester reaction remain unchanged. And that's consistent with this model of the sole role of these two residues to be donating hydrogen bonds to the O2 oxygen. Fanny also mutated this N100 active site residue to alanine and saw a similar or largely equal effect in the rates of hydrolysis of both substrates. And this is presumably because N100 donates a hydrogen bond to this O1 oxygen, which is common to both substrates. So all these data are consistent with the structural prediction. And we can actually leverage this now as a mechanistic tool. We can use this feature to ask how many of the catalytic effects all over the enzyme are functionally coupled to either R164 or K162. So to give you a more specific example, let's consider this second shell residue, Y112. Y112 has a catalytic effect, has a 12-fold catalytic effect when we mutate it either to glycine or valine, and it sits directly adjacent to R164. So we expect that Y112 might be functionally coupled to this residue. So we test it simply by mutating Y112 and assaying both substrates. And if we see a drop in monoester hydrolysis, but no drop in diester hydrolysis, we know that it's functionally coupled to one of these residues. If we're wrong, and Y112 is instead functionally coupled to N100 or the zincs or another active site element that interacts with a part of the substrates that's common to both, we expect an equal drop in the rates of both substrates. So we did this experiment for all of our mutants. We assayed the rates of both monoester and diester substrates and compared them and discovered that 156 residues, 156 mutants with catalytic effects had FC1 effects, meaning that these residues are somehow functionally coupled to K162 and R164. And if we look at the biggest effects, unsurprisingly, we see that most of them are clustered in the second and third shells around these two residues. So the K162 and R164 residues are shown in pink, and the FC1 effects in the second and third shells are shown in spheres around them. And we can see, as expected, this Y112 residue is functionally coupled to these active site elements. So It's important to note here that while we may have expected that the second shell, the things around the active site residues we're interested in, are going to have effects, there's no way we could have predicted which of the residues, which of the 10 had the functional effects, nor what their magnitudes would be. And what we really couldn't predict was that not only are these effects limited to the second shell, they actually extend throughout the enzyme all the way to the enzyme surface. So for example, there's this D473 mutant, which is on the surface of the enzyme, which has an FC1 effect of magnitude that's very similar to the second shell residues. And this is telling us and suggesting that these surface mutants may be providing opportunities for engineering allosteric control of individual catalytic mechanisms. So PafA is not known to be an allosteric enzyme by any means, yet we see these surface patches of functional effects that are somehow functionally coupled to specific elements of the active site 20 angstroms away. And in the future, it's going to be really exciting to use HT-MEK to sort of drill down and test models for how these surface sites are functionally coupled to the active site. Now, the second functional component I'm going to tell you about concerns a completely different mechanism of catalysis used by the enzyme. So remember that enzymes need to evolve to be specific for the transition state. That's going to give us an enhancement in the rate of reaction. We also want to limit interactions with the ground state, because if our ground state interactions are too tight, this means we can end up saturating the enzyme at low substrate concentrations, and also can have the unfortunate consequence of making the enzyme very susceptible to product inhibition. So we know from prior work that PafA and related alkaline phosphatases use a ground state destabilization mechanism. So in the ground state, which I'm showing schematically here, the negatively charged 3' nucleophile is sort of pushed up against the negatively charged phosphoryl oxygens, resulting in a lot of electrostatic repulsion. Now this repulsion is relieved in the transition state because when the nucleophile attacks and this bond starts forming, we change to a partial negative charge and reduce the interaction, or reduce the repulsive energy. And so we can actually measure the effect of mutations on this ground state destabilization mechanism by simply measuring the affinity of a ground state analog, which is phosphate. So imagine that we make a mutation that sort of moves the nucleophile away, or allows it to move away from the substrate. That gives it an opportunity to relieve this electrostatic repulsion by, instead of reacting, by just simply moving away. And what we expect is that this will actually result in tighter ground state binding and tighter phosphate binding. And so we measured the affinities of all of our mutants for inorganic phosphate in our library on the chip, and consistent with this expectation, we do see mutants that significantly tighten the affinity of the enzyme for phosphate. So for example, there's this H83G mutation, which increases the phosphate affinity about 100-fold. And we actually did all of our mutants and identified over 200 mutants that tightened phosphate inhibition significantly, indicating that the ground state destabilization mechanism depends on extensive scaffold interactions. And as with the FC1 effects, we see that there's lots of effects around the active site and around the nucleophile, as we expect, but these actually form only part of a larger contiguous region of effects that extends from the active site throughout the enzyme, again, all the way to the surface. And if we compare FC1 effects with our FC2 or our ground state destabilization effects, we see these regions that are spatially distinct from one another. And this is telling us that specific regions running throughout the PafA scaffold are important for individual and distinct catalytic mechanisms. So we can see that most of our FC1 effects sort of fall on the right here around our two active site residues, K162 and R164, while all the ground state destabilization or phosphate binding effects sort of wrap around the other side of the enzyme. And again, in future, it'll be really interesting to dig down into these networks using higher order mutant cycles with HT-MEK. Now, in the final few minutes, I want to tell you about some unpublished results that I think are really interesting. So we not only measured catalysis for multiple substrate and the affinity of all these mutants for a ground state analog, phosphate, we actually measured their affinities for a transition state analog, tungstate. Now, tungstate has been established as a transition state analog of PafA and related enzymes by people making all of the active site mutants and seeing for any active site mutant where you see, for example, a 10-fold drop in catalysis, you also observe a 10-fold drop in affinity for the transition state analog. So it's the correlation between catalytic effects and transition state analog binding effects that sort of allows us to define tungstate as a transition state analog. But when we do this experiment for our entire library, we see something kind of strange. So remember, we expect that our transition state analog affinities should decrease concomitantly with our catalytic effects. So all of our mutants should fall on or about this one-to-one gray line. And indeed, that's what we see for the active site and second shell residues, shown here in red and orange. But for the more distal residues, we actually see something completely different. We see lots of catalytic effects that have no change in the affinity for the transition state analog. And we actually also see a whole bunch of mutants that are down in catalysis, but for which the transition state analog affinity actually tightens. And this was completely unexpected. So here's what we think is going on. And this is just a sort of schematic. So let's imagine wild type enzyme and sort of look at schematics of the transition state and the transition state analog binding. Now let's imagine in wild type that all these interactions in the transition state are highly optimized. So for example, R164 and K162, are precisely positioned to interact with this O2 oxygen. And let's imagine our transition state analogs, we know are not perfect recapitulations of the transition state. For example, the bond here between the oxygen and the tungsten atom is probably a little bit shorter than the bond forming in the transition state, and the axial equatorial bond angles and the bond lengths are probably a little bit different. So things are not going to be perfectly optimized. So let's imagine that our K162 residue can still donate a strong hydrogen bond to this oxygen, but R164 is a little bit mispositioned to be forming a really good interaction. Now let's imagine we make a distal mutant. So we leave our active site intact, but we make a mutation a few angstroms away. And imagine that there are some conformational rearrangements that somehow end up bumping R164 a little bit. And so in our transition state, we've gone from being very precisely and ideally positioned to maybe being a little bit too close with a steric clash, or it could go the other way, make a slightly worse interaction. But fortuitously, by making that conformational change away from the active site, we've actually pushed we've pushed R164 into a conformation where it might favorably interact with the transition state analog now. So it's a worse catalyst, but a better transition state analog binder. And so we're in the process of doing these experiments not only for tungstate, but also vanadate and arsenate, which are other inhibitors from the enzyme, to see if we can map or use this information to understand the changes that might be happening in the conformational ensemble at the active site when we make more distal mutations. So in summary, what I've showed you is that we've started with catalytic effects, and then sort of like one of those Russian nesting dolls, sort of uncovered more and more features of PafA function by having the ability to measure multiple functional parameters. So we started with simply Kcat over Km for a cognate substrate, and then identified that a lot of these mutations result in a change in the active fraction of enzyme, as well as catalytic effects. And then we further dissected our catalytic effects into effects on individual catalytic mechanisms. So for example, FC1, which we use specificity, substrate specificity to read out, and ground state destabilization, measured by tightening phosphate inhibition. Now, I didn't have time to tell you about our other two functional components, but we also used phosphate binding, weaker phosphate binding, to identify mutations responsible for preferential transition state stabilization in the enzyme. And we also identified not only mutants that had catalytic effects, we actually identified mutants that affected the rates of an individual microscopic step of the catalytic cycle. In this case, hydrolysis of the phosphoryl enzyme intermediate, and refer to that as functional component 4. Now, just in the final minute or so, I'd like to just summarize the conclusions of this talk, but also talk about some of the future directions of HT-MEK. So the first thing is that HT-MEK works. We can now do enzymology on the scale of thousands, rapidly and inexpensively. And in principle, although we've only really done PafA so far, we should work for any enzyme with a fluorescent readout, either direct or coupled. So you can imagine doing proteases, glycosidases, but also things like metagenomic variants to understand how sequence changes in the environment might have differential effects on all these functional parameters. We've also seen that PafA function is modular. So active site elements are functionally and extensively coupled to their surrounding residues, and not only their surrounding residues, but throughout the larger enzyme. And if we look at our FC1 effects, for example, we see that most of our effects are in the second shell. And if we draw these out, we can start to hypothesize interactions between the second shell residues and the active site residues, as well as between the second shell residues themselves. And it's this context that I think we're missing when we try to design enzymes. So when we try to design enzymes, I think we're at a point where we know pretty well what reactive groups we want to put at or near the active site, but how to build sort of a mold or a scaffold around them, i.e. how to build the functional context, is something we don't know how to do as well. And it's data sets like this that ultimately will improve our knowledge. We've seen that individual functional effects extend throughout the enzyme to the surface. And in future, I think this is going to provide a lot of opportunities to identify allosteric sites and also map allosteric networks to understand how allosteric functions. Because PafA is not an allosteric system, yet it has these surface sites. And having these surface sites, maybe this is a more common feature of enzymes and proteins than previously thought. And maybe we can identify these using HT-MEK and then engineer mechanisms of allosteric control, sort of by highlighting what surface sites to bind molecules at. Something I'm particularly interested in is further characterizing these long-lived kinetically trapped misfolded states. So we've identified all of these mutations that somehow bias the folding trajectory of PafA either towards the active folded state or an inactive misfolded state. And it'll be really interesting to try to understand what are the functional interactions that are sort of guiding PafA along its folding pathway. And can we do this for both PafA and other proteins and learn general rules or general mechanisms of protein folding? And having all these misfolded mutants actually provides an interesting opportunity to maybe study chaperones. So for example, you can imagine doing an experiment where we take all of our misfolded mutants, our misfolded library, for lack of a better word, and adding in chaperones and seeing if any rescue occurs. And because we have the phosphate sensor, we could be doing these experiments. We could be measuring chaperone function by simply reading out whether we rescue PafA mutants via PafA catalysis, but simultaneously be looking at hydrolysis in the ATPase domain, for example. And so I'd just like to close by acknowledging my collaborators and co-authors who we worked closely with and were essential to this project. So for example, Daniel Mokhtari, my co-author, with whom I worked closely over sort of all aspects of designing, optimizing the technology, as well as making the measurements. Fanny Sundin, a super talented research associate in the Herschlag lab, who actually did most of the prior characterization of PafA. So we're really standing on Fanny's shoulders when we move away from the active site to study the entire enzyme. And Fanny also did most of the characterization of the misfolded state. Mason Appel, a postdoc, and Scott Longwell, a graduate student in bioengineering, they were essential for helping us acquire data as well as making significant contributions to automating parts of the assay and improving the process of acquiring and analyzing data. And then of course, my mentors, Dan Herschlag and Polly Fordyce, who I think everyone here knows. And I was actually Polly's first postdoc, and I just feel, really privileged to have been around and just watched her build up her lab to this real epitome of success. I feel really privileged for that. And then all of the lab members who I didn't have time to acknowledge personally, but who nevertheless make the environment great. And lastly, I want to thank everyone here for giving me this opportunity and to present this work. And I'd take any questions. Thanks a lot.
Thanks, Craig. That was, even hearing the third time, it was really great. So we have a couple of questions, so I just want to start with that. So the first question is from Subu. Hi, Subu. So he asks, did you try urea titration? Maybe 2 molar urea is too low to destabilize the mutants.
Right, so we did 0 to 2 molar, and we could have done a full urea titration, but we didn't want to for a couple of reasons. Because remember, we have this surface immobilized enzyme, but none of those interactions are covalent. So we didn't really want to get into the fact if we add maybe 6 molar urea, we just dissociate all of our protein and can't measure it. And again, we're only interested in the mutants that are highly destabilized at low urea concentrations. So if you're not significantly destabilized under standard conditions, the stability might have changed, but that's not going to give you a catalytic effect under standard conditions. And so we sort of didn't worry about sort of subtle changes to stability for the other mutants. We were only worried about, do any of our observed catalytic effects arise from equilibrium unfolding? Now, some other people in the lab, especially Bia here and Fanny, are actually working on an orthogonal assay to measure protein stabilities on chip more sort of accurately and over the full range. So hopefully that will sort of be out soon. But for our purposes, it's sort of sufficient to only make sure that none of our catalytic effects are due to this equilibrium unfolding process.
Okay, so I'm going to ask Cody's question now because I really like it. So Cody says, really cool talk.
Thanks.
So he's asking, what other readouts could HT-MEK be adapted for in the future? And also, was there any pattern in the identities of the residues in the second shell and further that have catalytic effects?
Both great questions. So basically, any enzyme that can generate a fluorophore should be amenable to HT-MEK. So there's one problem I didn't have time to tell you about, and that's that the PDMS chips themselves are super hydrophobic. So what happens, and actually this is a problem throughout sort of the field of microfluidics, if you have a hydrophobic molecule, they tend to diffuse into the PDMS over time. So you can imagine situations where you build up product, but then you have a process on top of that that's sort of removing your product from solution into the chip. So actually we ended up synthesizing two novel substrates based on some commercial substrates, because the commercial substrates we used, although they reacted, they ended up diffusing into the ends or into the chip, and that limited the dynamic range. So we ended up having to synthesize with some charged groups on the fluorophore, which didn't change the fluorescent properties, but kept it in the solution and not the chip. So that's sort of a limitation with using fluorophores. But I think the fact that we can do these measurements with the phosphate sensor get around a lot of those problems. So now with the phosphate sensor, phosphate's very negative. It doesn't diffuse into the chip at all. So any enzyme that you can generate phosphate, you should be able to, in principle, measure with HT-MEK. And, you know, that's either directly or via a coupled assay. So sort of the world's the limit, and you know, we want to start testing more and more enzyme systems to really truly find the limits of what we can and can't measure. But I think it's going to be pretty applicable to a lot of things. And so the second question was, do you see any sort of like features, common features in the second shell in these regions? We actually don't, but we need to look more extensively. So we see these regions. In the regions, I didn't emphasize this, but we see similar regions when we make valine and glycine mutations. So that's sort of telling us that, it's telling us in the sense that it's the wild type residue that matters. It's not solely a function of putting in a different residue. And, you know, we see hydrophobic residues that have effects. We see hydrogen bond donors and acceptors that have an effect. So there's nothing obvious, but it merits a further look. And I think as we get more and more data on more and more systems, we'll be able to do more of those comparisons.
Okay, Tanya, do you want to ask your question?
Yeah, really impressive work.
Thanks.
Really enjoyed this. Makes you think about lots of things. Fantastic. So I have many questions. Hopefully we'll have time later. I guess one thing to me, one of the most surprising findings was the considerable fraction of mutants that have these long-lived kinetic trap misfolded species, right? And so I'm wondering what you think about that observation in terms of being general, perhaps, or something that might be rather specific to the system that you picked here because it is so stable and also, you know, diffusion limited and so efficient.
Yeah, so to us, we didn't expect it at all. So I'd be curious to talk to other people and see. Some people may have expected something like this, but we definitely didn't. So we went in thinking that we might have equilibrium unfolding effects, but didn't really occur to us that we'd have these misfolded states until the data led us there. And I think you hit the nail on the head when you said that it might be because PafA is super stable. So PafA is a super stable enzyme. You know, you can sit it in like a high concentration of urea for like 2 weeks without any perceptible change in activity. And maybe one consequence of folding into a very stable state is that you become more prone to forming very stable misfolded states. If you take a protein like DHFR, which is well known to sort of equilibrium unfolded in a two-state transition, neither of those wells are ever that deep. So maybe you just can't, there's no real sort of force, there's just no way you can like find a solution that gives you a kinetic trap for a system like that. But if you start with a system like PafA, which has a deep folding well, you might be, because of that, as you point out, prone to these misfolding wells. And that's sort of, yeah, I think it probably is more general than we think. And I think maybe we can use PafA as a system to start to understand the molecular determinants that might be responsible. So we suspect PafA is so big, you know, we think it's folding co-translationally. So as it comes off the ribosome, you know, it might want to find specific contacts early, get you into an early folded state, and from there you're sort of okay to go. You can sort of wrap around and keep forming the right contacts. But early on, let's say you make a mutation and you disrupt one of these essential contacts, maybe that results in the formation of a sort of metastable early state that's wrong. And then as you start wrapping more chain around, you just sort of are always forced to go sort of down the wrong path. And I don't think we have a great understanding of how that process happens. And so I think that'll be definitely really interesting to follow up. And then you can imagine adding chaperones and things like that to see if you can sort of either stabilize or rescue it during this process. And I've sort of started trying some of those experiments, but it's still early days. But I think it's a really cool question.
Okay, so let's try and squeeze in one of the second Suba's question, and then we can wrap it up. So Suba asks, I assume you have measured the contribution of the residues to FC1, FC2, and so on. Mm-hmm. Do residues cluster by their FC contribution, or is it the case that a subset of residues end up contributing to all the FCs?
They generally cluster, yeah. Clustered probably more than we thought going in. So this is probably the best slide that shows that, but you can see the FC1 effects cluster around these two residues, K162 and R164, sort of coming towards us out of the screen, whereas the FC2 residues form a very contiguous region, but on the other side of the enzyme, and they're largely distinct from one another. So mutations that affect ground state destabilization don't tend to have FC2 effects, and vice versa. And I didn't have time to show it, unfortunately, but there's a whole bunch of mutations on this green helix that only affect phosphate inhibition. They have no other functional effect. So this is telling us that the function is pretty modular.
Okay, let's all quietly in our rooms thank Craig again. Thank you everyone for coming. So if anyone is really keen on talking to Craig and you don't have a meeting with him, just shoot me or Gina an email and we can make it happen.
Yeah, please do. And thanks again, and thanks for listening. So it's really great to present this work.
Yeah. Well, okay, I'll talk to you later. Good, have fun talking to other people.
Thanks.
Bye.
Yeah.
Bye.